So genetics 1, what we're trying to understand here is that every organism is, or yeah, we'll go with every organism that has, like mammals, for example, are going to have 2 sets of their chromosomes, and one that they get from each parent. We call that being diploid. In order to reproduce, we can't just have both parents pass on all of their chromosomes to their offspring, because then they go from having 2 copies of each chromosome, and therefore 2 copies of each gene, to having 4. And so we need to find some way to take that number and reduce it down. And that is what meiosis is. But we don't want to just have a random half, right? We can't have offspring get 2 number 1s from one parent and no number 9s from that parent, right? We need to have each parent pass on one of their 1s, one of their 9s, one of their 16s, one of their sex chromosomes. And that would make, if they pass on 1 each, that would make it so their offspring end up with the full sets, 2 of each chromosome, just like the parents had. So we need a way to divide up our chromosomes and have that happen. Now, when we say they're in pairs, right, that means that this chromosome and this chromosome, we call them homologous pairs, they have the exact same genes on them. Now, the genes are the same, like say this was the gene for eye color, right? It would be on both of these chromosomes. But this gene for eye color might be blue eyes here and brown eyes here. So the alleles can be different. So these are homologous chromosomes because they have the same genes, but they are not sister chromatids because they're not exactly identical. Even if they were, we wouldn't call them sister chromatids. We'll come back to that term in a minute. All right, so diploid is when you have 2 copies of each chromosome. Haploid is when you've been reduced it down to 1. So when we make a gamete, a sperm or an egg, we want to go from being the diploid organism that we are to a haploid organism, from 46 chromosomes in humans to 23, but not a random 23. We want 1 of each type. So that is kind of the basis for what we're looking at here. Okay. Now, these 2 videos do a good job discussing the division process, but I just really wanted to kind of give you a brief overview myself here. Okay. So in meiosis, we have 2 divisions, meiosis 1 and meiosis 2 depicted here. Before this process begins, our DNA is going to be copied. Every chromosome will make a duplicate. Those duplicates will still be attached to one another. So even though our chromosomes have been copied, they're still considered one chromosome. So we have 1 chromosome to start, we copy it, and it's still considered to be 1 chromosome because it's still that singular structure, right? They're still attached. Now we have 2 copies of every gene in those 2 chromosomes. Now, once meiosis begins, this copied pair, we call these sister chromatids that form 1 chromosome, will go and match up with their homologous pair, which is also copied. So we'd have 1 set of arms and the other set of arms is kind of the idea. And they're going to be paired together. And it's going to be the set that you got from your mom and the set that you got from your dad. And that's what it's depicting here, right? So we have 6 total chromosomes. So 2n equals 6. And then we are going to take those homologous pairs that are paired up, and we're going to line them up along the equator. And then we're going to pull them apart in anaphase, so they go to the different sides. And then telophase and cytokinesis, we're going to form new cells there. Okay. Then it's going to go through the process again, but this time it can't line up in homologous pairs because now the homologous pairs are in 2 separate cells. So those cells are going to line up along or those chromosomes are going to line up along the equator of the cell. This time end to end rather than side by side like we see here. And this time we're going to separate the sister chromatids. So here we separated the homologous pairs that are copied. And then here we separate copies, the sister chromatids, to each side. So those two divisions will end up with having cells that end up having 1, 2, 3 chromosomes, 1, 2, 3 chromosomes. So now instead of 6, we end up with 3. And so we went from being diploid to haploid. Are there any questions on meiosis? The purpose of meiosis is really about reproduction. It's different than a process called mitosis, which we will cover next week. But there's some similarities, but this is the one we need to know right now to understand genetics. Now, Gregor Mendel comes up with 2 laws that describe genetics. The first, he didn't know what DNA was, it hadn't been discovered yet, so he described it as a heritable factor. He said, the heritable factors must segregate. So he gives the law of segregation. The heritable factors must segregate. What that means is mom might have given you the recessive allele, and dad might have given you the dominant allele. That means you're heterozygous, right? Those are the 2 factors that he was talking about, the 2 alleles. What that's saying is that this parent can either give you the recessive allele, which will end up in this cell, or the dominant allele, which will end up in this cell. They can't give you both. They can't give both of their alleles to you. And so the reason why is because of this part, right? Metaphase and anaphase 1 and meiosis are showing us that segregation of the recessive allele going into ultimately into one cell, and the dominant allele ultimately going into the other. So that's the law of segregation. He also talked about the law of independent assortment. And that would mean if we had a second gene that's here, let's say mom gives you the dominant one this time and dad gives you the recessive one. Well, how they line up is completely random. So in this case, the recessive and the dominant lined up with the dominant on the bottom and recessive on top. It's completely random. It could have lined up the other way. And in that case, both the dominants would have gone to the upper one and both the recessive to the bottom. But because of how I described it there, dominant and recessive, and over here is recessive, dominant, and this is recessive. So recessive and dominant, recessive. Well, one's going to go one way, one's going to go the other way. So this parent, this division would not pass on both dominants together. They'd be split. If they aligned differently, if this blue had ended up on top and the red had ended up on the bottom, then both dominants would go the same way. And so that's what Mendel was saying, is that that 2 characteristics are not tied together, right? The dominant and the recessive could end up assorting independent of one another. So these 2 traits don't have to be inherited together, essentially. And that was his law of independent assortment. I think it's super cool that without even knowing that DNA existed, let alone what a chromosome is or an allele, he described the process of meiosis, right? That these homologous pairs are going to separate, the 2 heritable factors must segregate, and the 2 genes that are found on different chromosomes are going to be inherited independent of one another. You can have both dominants, you can have 1 dominant, 1 recessive, 1 dominant, 1 recessive, or 2 recessives that they get to mix so they can be independently inherited. And that's why we have to, when we do a Punnett square, do all the different combinations that we can, because we're wanting to account for the ways that they could possibly line up right here in metaphase 1. All right. So that's the main thing from there. The videos honestly do a great job going over these processes and these laws. So make sure you check those out if you didn't.